The Clyde Collins Snow Forensic Anthropology Laboratory of the Forensic Anthropology Foundation of Guatemala carries out the forensic anthropological analysis of skeletal remains and all elements recovered during investigation in the forensic archaeology phase. The purpose of this is to determine a biological profile, individual features or characteristics, and cause of death, all of which will later aid in the identification of the deceased. But how do we do it? In many cases, the bones that may be covered with soft tissue or adhesive materials, such as grease and dirt, that hinder the process of analyzing the skeleton. The washing and labeling process refers to the procedure of cleaning and labeling both the skeletal remains and the associated artifacts. It begins by requesting the recovered skeletal remains and the exhumation file filled out in the field from the evidence warehouse, where the chain of custody of the individual remains will be filled out. The next step is a review of the X-rays from the case to be analyzed in order to have a broader understanding of the remains to be washed and reviewed prior to the washing stage. Any peculiarities, such as the presence of radiopaque objects or signs of violence, are recorded. The box of remains will then be opened for examination with the help of the exhumation file. If any relevant findings are observed before washing, it is necessary to take a pre-wash photo in the photo studio. The labeling should include a unique FAFG case number, a skeleton number, the grave where it was recorded, and a part or lot marking. The clothes are then immediately cleaned. This will be done by using brushes, with the aim of removing as much soil as possible. This process will lead to a search for elements such as metallic remains, clothing colors and/or patterns, signs of violence such as cuts or tears caused at the time of the victim's death, etc. Once the clothes are clean, the skeletal remains are gently washed with a brush and water, with the aim of removing as much soil and roots as possible. They shall be dry cleaned with a soft bristled brush to ensure their preservation and to avoid altering any signs of violence or individualizing features. Finally, the skeletal remains are labeled with a unique FAFG case number, a skeleton number, the grave where it was recorded, and a lot marker. The analysis process begins by spreading out the skeleton. The remains are placed anatomically on a table covered with a cloth, preferably blue in color. The main objective of osteological analysis is to determine the biological profile of a skeleton or skeletons. The analyst will use different anthropological methods to establish sex, age range, age group, or bone development stage, and individualizing features, such as antemortem fractures, defects, pathologies, and stature. Determining the biological profile is also important in the identification of the person, as it forms the basis for the comparison of antemortem and postmortem results from the forensic investigation. Once the bones have been anatomically placed, the condition of the skeletal remains will be established by filling out an inventory, which can be found on the analysis form. This allows us to discover the presence, absence, and state of preservation of the skeletal remains. In parallel to this, any reconstruction of anatomical regions required is carried out. This procedure is important because it helps to create a proper visualization of the damage caused by injuries close to the time of death, circumortem, as well as any postmortem damage. After completing the inventory, the events that affected the body after death, for example, taphonomic processes, will be documented by means of a skeletal graph that creates a general visualization of these findings. To determine the sex of an adult skeleton, two general elements of the bone anatomy are observed: the pelvic girdle and the skull. On the pelvic girdle, 9 particular regions located in the pubic region are examined, while 6 features are observed on the skull to determine sex. The analysis of both regions makes it possible to determine whether the analyzed remains correspond to a male or female individual. The biological age range in adult skeletons is established using 6 methods, comprising the analysis of the development stages and degenerative changes in the pubic symphysis region, the sternal end of rib 4, the development of the periodontosis, and the transparency of the dental root, and finally the development and stage of fusion of the vertebral rings. To estimate the height of the skeleton, a method based on the measurement of the maximum length of the femur or tibia is used, which is obtained through the use of an osteometric table. Obtained are processed by a formula that provides a height range. This range cannot be estimated in young individuals because the epiphyses are not fully fused. Determining individualizing features includes looking for evidence of trauma that occurred during the victim's lifetime. The most common finding is a bone fracture with evidence of scarring, represented in the bone callus seen in the images. Any other pathology is also recorded. For example, any anatomical and physiological disorders that could have affected the victim's life, manifesting themselves in symptoms as well as any visible defects or pathology in the bones and dental pieces. Finally, a thorough examination of the dentition will be made, with the aim of finding pathologies, defects, dental work, and any particularity that can be described as an individualizing feature of the victim. The determination of circumortem trauma, for example, injuries that possibly caused or contributed to the death of the victim, is an important part of the analysis performed in the laboratory. Also called perimortem trauma, this occurs around the time of death and is characterized by an absence of scarring. The analysis should record and describe each trauma observed on the skeletal remains, and then interpret the pattern of injuries, trajectories, and their relationship to other traumas. Finally, the description of traumas will be complemented with drawings and photographs of each trauma observed. At the end of the analysis stage, the data obtained will be entered into the individual report. This consists of a digital standardized template. Once this report is complete, the analyst will request for a photographic record to be made. The purpose of the photography process is to create a visual record of the findings observed during the analysis phase, which can be used throughout the investigation. The visual record includes photographs from the pre-washing stage, of the spread out skeleton, and of the study, which includes the skeleton's clothing, associated items, protective materials, dental records, biological profile, individualizing features, circumortem trauma, and skeletal samples. For each of the photographs, color codes and metric tokens are used to identify the different findings during the analysis process. Blue is used for sex and age determination, green for defects and pathologies, yellow for antemortem trauma, red for circumortem trauma, and white for dental records, metal remains, labels, coins, and skeletal samples. In the Department of Forensic Anthropology, 3 types of skeletal samples are taken. The MG01 genetic sample corresponds to the first sample taken after the skeleton arrives at the evidence unit. Samples MG02 and/or MG03 are taken until the end of the analysis process. For sampling, a half-letter manila envelope, tamper-proof seal, craft paper, white scale, and electric saw must be prepared. With the equipment ready, the scale and manila envelope are filled with the sample's nomenclature, the date of the sampling, and the signature of the person in charge. For sampling, priority is given to dental pieces in a good state of preservation, for example, without any severe caries, defects, or pathologies, or with dental warp. Before taking the sample, a photograph of the bone or anatomical region to be sampled is taken, prior to the cutting or extraction of the dental piece. The cut or extraction of the dental piece is then carried out, and a second photograph is taken of the bone next to the fragment that was cut, to indicate from which region the sample was taken. The same procedure is carried out for dental pieces. To finalize the record, a third and final photograph is taken of the isolated bone fragment or dental piece. Once the sampling process has been completed, the sample is packed in a manila envelope with the tamper-proof seal affixed. The date and exact time of sampling should be noted on it. A chain of custody form should be filled out for each of the samples taken, declaring the sample's nomenclature, the name of the exhumation site, the name and signature of the person responsible for taking the sample, date, time, sample description, and one of the photographs taken should also be attached. The sample collection process ends with the delivery of the record to the forensic genetics department. They will then deliver a copy of all documents received with their respective stamps to be scanned and delivered to the evidence warehouse. Once the analysis process is complete, the skeletal remains and associated items will be packed. The box is sealed with evidence tape, dated and signed by the analyst, and then returned to the evidence warehouse. Some additional processes are then carried out in the laboratory, including the description, measurement, and photographing of evidence and recovered metallic remains. The latter will also be weighed and packed in plastic canisters with their respective nomenclatures.